So Dr. Redline, are you ready?
I am ready. Thank you.
Fantastic.
Okay, and I apologize for staying off camera, but I'm having a finicky internet connection right now. But I did want to very much thank our hosts for having us today and everyone attending. We're very excited to be talking about sleep. Next slide. So what I will be doing over the next period of time is to give you a very brief overview of what is special about sleep and the excitement of having sleep be part of NHLBI data resources. And I'll provide an initial overview of NSRR and some of our approaches to data sets and metadata harmonization. And then my colleague, Dr. Sean Purcell, will actually share with us some of our approaches for developing and sharing data and sleep analysis tools. Next slide. To begin with, sleep is I think maybe for those who you're on the call, you're already interested in sleep, but I wanted to emphasize that there is growing scientific as well as public health and clinical interest in the vast importance of sleep as a crucial pillar of health as it relates to nutrition, physical activity, and sleep management. And even the American Heart Association, as you could see by the bottom graph of Life's Essential 8, in just this last year, added sleep health along with 7 other components like diet, activity, blood pressure control as really critical to maintaining good cardiovascular health. Next slide. And not only is sleep integral for good health and actually interact with many of the phenotypes and disorders within biodata catalysts, but there's very exciting data about the importance of circadian biology and circadian rhythms. As you can see here is just a hypothetical biological rhythm in health in blue, and maybe how it might change in red with disease or aging. And we now know that not only do we have there's a critical role of our central circadian clock in regulating multiple biological rhythms, but there's also many interactions with our central clock with many peripheral clocks that are in most of our tissues. So again, the really growing excitement of understanding both sleep and circadian biology as critical biological mechanisms and components of health. Next slide. But when we talk about sleep, and this is very relevant to those of us interested in sleep data, it's very important to note that sleep is not a single unitary type concept, but sleep and circadian health can be described across multiple domains. And these may include sleep duration, sleep timing, rhythms, fragmentation, efficiency, depth, measures from brain EEG recordings on microarchitecture, macroarchitecture, clinical definitions of sleep disorders, and the patient-reported outcomes that describe daytime sleep sequelae. Next slide. And each of these domains may be measured by a variety and often quite different types of instruments, each of which really impact the types of data that we have available in our approach to visualizing, annotating, and analyzing the data. So for example, EEG-based metrics of sleep microarchitecture, sleep apnea often require a sleep, overnight polysomnography studies. Sleep-wake rhythms, on the other hand, may use, wrist-worn wearables or activity trackers. Issues about sleep, metabolic changes may use imaging data, and patient-reported outcomes will use various surveys. Next slide. And one of the really unique aspects of sleep data as we think about biodata catalyst and approaches of the National Sleep Research Resource is that sleep presents really maybe more granular and more temporally informative data than almost any other data domain. In fact, when we collect data overnight using polysomnography, we collect multiple, that's poly in the polysomnography, multiple physiologic signals over many hours of data collection. When we collect actigraphy or wearable data, we may collect data over days, weeks, or even months. And the collection of these multiple streams of data and over time provide many, many exciting data opportunities, but also data challenges. Typically, much of our research has taken these very rich data sources and collapsed them to very simple summary metrics. However, advances in our thinking and our toolbox now allows us to extract, very much more informative features. For example, the bottom of the slide, measures of hypoxic burden, or on the top of the right-hand side, quantitative measures of spectral EEG spectral analysis overnight. And these, again, provide opportunities for us to think about ways to share data and ways to utilize data. Next slide. And sleep data has some issues that are common to other data that are in BDC and other repositories, and some unique issues. As with other data sharing, data access, there are issues of ownership and regulatory type issues. But specifically for sleep, and although it's not unique to sleep, it is a feature we have to think about. Often, whether it's research data sets or clinical data sets, the data we collect is often heterogeneous. That is, it may be collected using a variety of equipment, what we call signal montages, or really approaches for how the data is filtered and displayed and sampled. It may then have different layers of human manual scoring attached to these. And the extent to which we understand those variations also varies. Sometimes they're very well annotated and other times poorly annotated, all of which give challenges to standardization, harmonization, the ability of any user to access these appropriately or combine the data across multiple data sets. We also have some special challenges that NSRR specifically is eager to address and has been working on, about the development of standardized terminologies and vocabularies and analysis tools, as well as the reaffirming needs to keep documentation consistent and clear. Next slide. So what does the National Sleep Research Resource do? We actually started in 2013, and in the last 4 years, we've been supported by an NHLBI contract to specifically provide a community resource to overcome some of those challenges, as well as to take advantage of some of those opportunities of rich, temporally specific multi-channel data. And so it's a way to deposit and access sleep data, including those rich physiologic signals, including polysomnography and actigraphy and specialized sleep questionnaires. But it may include, according to the data set and the contributors' data restriction, a whole host of other data, including anthropometry, health behavior, physical activities, blood biomarkers, and so forth. And particularly one opportunity that integrating NSRR data and BDC data is the opportunity in many of the data sets to link our rich phenotype data with genetics data, and potentially in some cases even advanced imaging data. NSRR has also been recently ingesting data on sleep from animal experiments, as well as really complex circadian data. So those also are exciting areas. And as you'll also hear from Dr. Purcell, we're very committed to both develop and disseminate and document open source tools and support the sleep research community in terms of their engagement. Next slide. And we also have the NSRR platform itself also is committed to help users browse, search, and download the sleep data, and also help them with understanding differences between cohorts and accessing harmonized terms. Next slide. So when we think about a repository, you could think of it as a collection on the left of valuable, well-curated data sets, or if you go to the right, you could also think of a repository as really driving discovery and really supporting innovations in the research community. Next slide. And in this realm, our goal has always been to not only be a collection of data sets, but to add value and stimulate novel ways to think of analyzing sleep data. And we do that through improving over the original data we usually get of the documentations, providing search capabilities, doing certain amounts of harmonization and standardization for core terms and signals, providing metadata, improving, really adhering to fair and trust principles, developing and sharing open analysis tools, providing exemplar applications for our data, linking and integrating with other resources, including BDC, and building our community, including outreach and education. Next slide. So we've developed within the NSR itself mechanisms for fairly ready data access. And in fact, we have an online data use agreement that's fairly straightforward to complete, and that doesn't require an ERA Commons account. And because we have the data in NSR is not as sensitive as other places. We do not have genetics data, for example, and it's approved in general by our local data access committee. But we also have tried to encourage fairly straightforward and low burdensome sharing of data from the community with NSRR. And the process is outlined here. And for time's sake, I won't go into a lot of detail, but to say that we have a page on our website about data sharing, and it begins with really initiating a relationship with a potential contributor. We have some well-developed forms that contributors can access and complete to tell us about any limitations, scope of their data, describe the format of the data. We then meet with our contributors. We initiate a regulatory approvals, which does require a data use agreement just to share the data and begin to understand really what those data sets look like. We then begin the sharing data process, and we often will we'll review initial samples of data and work with the contributors that way. We will ultimately, as I will show in several slides, we will then add that value to the data, reformat it, and annotate it and share what we've done with our contributors to make sure we were faithful to the purpose. And then continue to get solicit feedback from the community on the clarity and the usefulness, and make quality improvement changes over time. Next slide. So where we are now, we have 26 data sets with another 8 to 10 data sets on the launching pad, just in really final stages of regulatory approvals and data modeling. These 26 data sets that we have now, though, include almost 50,000 individual overnight polysomnogram studies, about 30,000 that do have EEG studies, about 7,000 multi-day actigraphy studies. We've harmonized over 14,000 or annotated over 14,000 terms, including about 5,000 with detailed provenance information. We have about 5,000 data use agreements that have been executed, and we regularly share about 2 terabytes of data a week with the community. Next slide. Now, this is just a snapshot of our cohort matrix, and you could go to a number of tags to get an at-a-glance understanding of each of the cohorts. And in this particular slide, I'm showing you a data matrix, so you could see the 26 or so cohorts along the left axis, along that left column. And then if you go rightward, what you can see is the different folders that describe categories of variables that are organized conceptually, say anthropometry or medical history or sleep questionnaires. And the greens tell us which types of data can be found in each cohort. I do also, I know this is quite small, but in the circles on the far left-hand side are several of the cohorts that you will find in BDC. You'll find maybe some flat, simple sleep data currently in BDC, but the NSRR data, next slide, will actually provide further a deeper granular data on those physiologic signals, as well as those added variables and annotations, et cetera, that I described before. So our goal is to work as partners with BDC and really add sleep-specific domain data that can be linked to the other data in BDC. And after I speak, or after Dr. Purcell speaks today, you will hear an example of the first data set that's been migrated, Cleveland Family Study, CSF, and how you can link those data now with the physiologic signal data to genetics data, all in one platform. Now, next slide.
Dr. Redline, before we continue, we do have 2 questions in chat. Would you mind answering them now, or do you want to wait?
Yes, I could go ahead. The 26 data sets are all in NSRR. Okay. So you'll have to go to sleepdata.org. And the molecular omics and imaging data are not in NSRR. They would be in BDC for the selective data sets. So for example, we will be migrating data from Sleep Heart Health SHHS, which includes data from Framingham and Eric and CHS and Mesa, and there will be those omics data, et cetera, in BDC. So that's the opportunity. And I will let the picture team, the next question will address later when you see that demo, okay? Next slide.
Yes, yep, no problem. We can keep moving.
Okay. And this now goes into, again, just the approaches that we take to really make that metadata, the data that describe the data, useful as well as consistent. And we've created a number of forms that, like a metadata intake form, we ask our contributors to fill out, but we assist them with that. And and then we receive data, that can vary from REDCap forms to study protocols to the consent forms. And then when you get to the middle of the slide, then we as a team will work at developing that study level metadata, file level metadata, and variable level metadata. And the reason we do this is to produce a number of outputs that we hope is useful to the community. And we want to hear from you to find out how useful it is. And it includes some README files describing these data, as well as structured metadata and version-controlled standardized data dictionaries that we develop. In addition to the primary data dictionaries developed by the originating contributors, we also provide tools and data to look at summary statistics by various metrics, as well as we harmonize core terms, including terms that are in TopMed and terms that are particularly salient to our sleep community across data sets. So we actually do a lot of mapping and tagging. And again, you saw an example of the cohort matrix, some visualization tools. We also have some Shiny apps to help people do various analyses and understand definitions and so forth. Next slide. And this is a little bit more about how we organize and do some of that detailed curation of metadata. So as I mentioned, all of our variables are sorted or organized in standardized folder structure to help with browsing. We work very hard on standardized terminology when often standardized terminology doesn't exist for sleep data, and we'll talk about that later. We also try to standardize the labeling of of all the metadata, including details about references and scoring rules and scoring interpretations. And for selected variables, we apply tags to really help with either machine learning type queries or to help with some modern sleep questions. So for example, we have a compositional coding approach for all the hundreds of ways one could define an apnea-hypopnea index. So a machine could read that and know which AHI a given study utilized. And right now we're in the process of tagging various variables to map to a conceptual framework of sleep health using Dr. Bicey's Are You Sated framework. And then for variables where we can find inferentially equivalent terms, we do also harmonize these. And I need to give a big shout out at this moment for Ying Zhang, our data scientist, who has led this particular initiative and has done amazing work. Next slide. Yes, the answer is the meta standards. We are constantly reading up about community-wide meta standards, and they are, we try to be compatible with as many standards as we understand. That's an interesting question in terms of which standards people use, but again, the principles and the way to find these and utilize these should make these broadly interoperable. Now, as I mentioned, NSR approaches to data processing are very much informed by the special types of sleep data that we take great interest in. And as you can see here, it includes actigraphy, the polysomnography surveys. Even for circadian, it will be including melatonin levels and other potential biomarkers. So again, a lot of our approach is to understand how these different data types differ, what their sources were. We often need to extract information, integrate them, and sometimes apply calibrations or transformations or other standardizations. Next slide. And here's an example. Actigraphy or wearable data, you might get it from a device like an actigraph, or you might get it from a device maybe potentially like a Fitbit. But we often use terms that have not been necessarily standardized consistently across devices and studies. So we begin with a conceptual model with getting input from our community members about what this device and this recording approach is intended to collect. So in this particular diagram, you see various metrics that identify specific data that are extracted from multiple day actigraphies. If you look at the big X's, recording start time, rest interval end time, rest interval start time, recording end time, you could see that you can identify 2 days, and we make it very clear in our data model what each of these, what's the difference, for example, between a recording start time versus a rest interval start time versus a sleep onset time, and so forth. And by identifying these clear intervals or time points, we're able to improve the consistency of the time, the sleep-wake periods that then are given annotations like wake after sleep onset, or sleep efficiency, or rest period, or sleep duration. So this is one example of how we use a data model to then inform our maybe core actigraphy harmonized variables. Next slide. And as I said earlier, we do try to get the user very ready access to all the provenance of, or the metadata related to each of the variables. And here is an example of an insomnia severity index, which is a standardized questionnaire. And you can see on the left side, you have the name of this particular variable, its label. There's a description of what it means, references. You could search for all variables articles in this data set that, that are under this. You could see the mathematical calculation. You could find out if it's an integer, and you could find some synonyms or tags. And then in this particular data set, you could actually see the distribution of who had what level of score. Next slide. Now I'm going to turn the presentation over to my colleague, Dr. Purcell, who will now talk about the tools and briefly engagement. Thank you.
Hi, so thanks, Susan. And organizers, please feel free to interrupt me if you need time for the second component of this meeting. So we've spent some time working to complement our data resources in NSRR with appropriate software tools. And unfortunately, there's a real paucity, I think, of open shared tools in the field of sleep, as nicely documented by my colleague Tamar Sofer and her colleagues in this in-press research note. So in context of what some call a crisis of reproducibility across many biomedical research fields, we conducted a literature review of all sleep methods and algorithm papers published in a particular year. The vast majority of relevant reports did not publish either code, an example, or any data. And to be blunt, if a method can't readily be used by others, it's hard to see what value a paper describing that method really has. And of note, of 5 articles that supplied all of the above, 4 were published in journals that asked for code and/or data, pointing to a clear role for journal editors in this regard. Next slide. So in my own work and with the NSRR, we've developed an open source tool, Luna, mainly focused on sleep EEG, but have made some additional efforts to try to document and share this, to add tutorials, vignettes, and tried to make the code base accessible via different means in order to share this as a resource alongside the NSRR data and facilitate others' attempts to reproduce our analyses. It's not always easy for small or moderate-sized research groups to do this, and this certainly is a work in progress in itself, but I firmly believe that something is usually better than nothing. Next slide. We've built an automated pipeline around this called NAP we use to process and harmonize PSGs, which we'll be instantiating in the context of BDC. There are some interesting decision points in how much to change the original data when doing this type of activity. We've ended up only fixing, hopefully, uncontroversial features of the data, such as labels and formats, that make the data easier to work with, but do not change the fundamental information. And beyond that, we attempt to, rather than attempt to fix things, we flag more subtle issues here. Next slide. We've also recently added this Moonbeam module where NSRR users can directly pull individual studies into the browser for interactive viewing and analysis. You can try this out yourself at the link remnonrem.net. But following from this kind of tighter coupling of code and data, this is really where we'd like, I think, to take a lot of this work moving forwards to support what I sometimes call a reference-based approach to the analysis of sleep data. Next slide. Back one. Oh. Correct, thank you. So that is, most analyses of PSG data are effectively self-contained in that new data are fed in, some derived metrics come out, but calculated only based on the current PSG. But following from other fields, including human genetics, I think there are at least 2 ways in which existing data can be used directly to contextualize and augment new data. So first, leveraging representative reference panels in whom the same set of metrics are being derived. One can imagine normalized, demographically adjusted centile scores for metrics, which can aid interpretation of new data. And second, if a reference database also contains other features that sufficiently track with the PSG features, those additional features can be statistically imputed via model-based prediction. So as shown here, automated staging, where manual scoring is the feature present in the reference panel, is one form of this approach, but you can imagine many others, including prediction of disease state or risk, and as I've done with colleagues in the context of schizophrenia and other neurocognitive outcomes. And so we're starting to make more progress along these lines, for example, incorporating some age and sex-based normalizations of the sleep EEG and methods to predict so-called biological brain age from the sleep EEG in pediatric samples. But I do think over time, these types of model-based analyses tightly coupling code and data from NSRR and other sources will really augment the value of a typical PSG or sleep EEG study. So watch this space. Next slide. So Susan said, we're very keen to build a community around these data and tools. So to that end, we have a blog where we give this kind of information, and we really solicit guest contributions as well. So if you have relevant work or you've used NSRR data and you think others would like to hear about it, please do contact us. We have a forum for questions and so on, and events and webinars such as this. And yeah, as Susan said, us hearing from you and getting feedback to tell us what's useful and what's not is really key. So please, either today or later, please feel free to do that. Last slide. So yeah, just acknowledgements. Obviously a large team of people here, and I think in the interest of time, I'll leave this up and pause, and we can move on. Thank you.